Hi everyone, as we move along the nervous system lectures, now we're going to be talking about the cells that form the nervous system. Like I said earlier, there are only two main types of cells in the nervous system, which is amazing, and there are going to be the neurons and the neuroglia. Let's talk first about the neuron. Here are the structures of a typical neuron. So when you think of a neuron, this is probably what you think of. And so we are going to have the cell body, which can also be called the soma or even the pericardium. Within the cell body, there are going to be these extensions, which are called the dendrites. Moving on from the cell body, we're going to have what we call the axon. At the end of the axon, we're going to have what we call the terminal button or the synaptic end terminal. And here is where you're going to have the synapse of this neuron with other neurons, for example, or with a skeletal muscle or with a gland. And when we talk about synapse later on, we're going to talk about these differences. But for now, these are the main structures that you need to know. With regards to functions of these structures, the dendrites, they're going to be important because they will be the ones that will receive a lot of the stimuli that's coming from several areas, and they will pass it on to the cell body. The cell body might also be synapsing, as we can see over here from another neuron. This signal is going to travel then through the dendrites, through the cell body, coming on to the axon. Within the axon, the beginning of the axon is what we call the axon hillock, which is the point of no return. So once this neuron decides that this information is important and it passes the axon hillock and starts going down the axon, that's it. The information needs to move forward. We can also see over here that we have an oligodendrocyte, which when we get to the glial cells, we're going to see that it's going to be a type of neuroglia. And because it's an oligodendrocyte, I know that this neuron is located in the central nervous system. And the oligodendrocyte is important because it produces myelin, as we can see over here, which is this lipid structure that will surround the axon. And the information will go down the axon and come down to the terminal button or the synaptic end bulb and synapse with, like I said, another neuron or a muscle or a gland. And the last structure that we can see are these spaces that are left by the oligodendrocytes that are called node Ranvier. And this is going to be important to transmit this information that's going down the axon a little bit faster. And we will see this later on. But just keep in mind the function of the node of Ranvier. The neurons, they're going to display this great diversity in size and shape. Actually, their cell bodies, they can range from a diameter of about 5 micrometers, which is basically slightly smaller than a red blood cell, up to 135 millimeters, which is barely large enough to be able to see with an unaided eye, which basically means that you're not using a microscope. Now, this pattern of dendritic branching is going to be varied and distinctive for neurons in different parts of the nervous system. A few small neurons, they're going to lack axons, and many others, they're going to have these very short axons. And as we mentioned earlier in this chapter, your longest axon is almost as long as you are actually tall, because remember how I said that it extends from your toes to the lowest part of your brain. And in this example that we mentioned earlier, look the size of this neuron. It goes from the tip of your finger, passing through the spinal cord and ending up right over here at the base of the brain. And then it will unite with another neuron, which is what we call the interneuron. Remember, a neuron between neurons, and then it will synapse with another neuron. Both structural and functional features are going to be used to classify these various neurons in the body, and structurally, neurons are classified according to the number of processes that are going to be extending from the cell body. Therefore, we can see over here the multipolar neurons. They usually have several dendrites and one axon. as we can see over here. Most neurons in the brain and the spinal cord are going to be multipolar, as are all motor neurons or efferent neurons. As we can see over here, the reds are going to be multipolar neurons. Therefore, we can say that multipolar neurons are going to be found in the central nervous system. Next, we have what we call the bipolar neurons, and as we can see, they have one main dendrite and one axon. And these are going to be found in the retina of the eye, in the inner ear, and in the olfactory area of the brain, meaning that these bipolar neurons are going to be part of the special sensory organs. Then we have what we call the unipolar, or also known as pseudo-unipolar neurons, are these sensory neurons that they actually begin in the embryo as bipolar neurons. And then as a result of the differential growth of the plasma membrane of the cell body between the axon and the dendrite, the two processes are going to be brought together, as we can see over here, and they're going to fuse into this single process that will divide into two branches that's going to be a short distance from the cell body. And both branches are going to have the characteristic structure and the function of an axon. Therefore, they're going to be long cylindrical processes that will propagate action potentials. However, the axon branch that extends into the periphery, it's going to be called the peripheral process. It's going to have these dendritic branches at its distal tip, whereas the axon branch that extends into the central nervous system, which is called the central process, is going to end in the synaptic end bulbs or the terminal bulbs. And the dendrites are going to monitor a sensory stimulus, such as, for example, touch or stretching, and then these impulses will propagate toward the synaptic end bulb, which can be located in the spinal cord or already in the brain. So let's look at these different types of neurons on this image over here. So the ones that we want to look for are the unipolar or the pseudo-unipolar and the multipolar. We won't find the bipolar because the bipolar is only located in the special senses organs. And here we don't have a special sense organ. Therefore, I said that the unipolar is what will be our sensory neuron. Therefore, the cell body is going to be located in the peripheral nervous system. So an example of the unipolar on this image is this green neuron. It will start over here on the tip of the finger. It will travel down, and the cell body is located right over here. Notice how there's a little bit of a little ball right over there, which is going to be representing the cell body. Now, this cell body is located in this enlargement area that's right next to the spinal cord, which is called the ganglion, or ganglia if you're talking about plural. Another thing that I want you to notice is the morphology of this cell, because this cell has the morphology of the unipolar neuron, where it will have a dendrite on one end, an extension of the axon, a cell body in the middle, and then another axon coming in with the terminal button or the axon terminal. So this is exactly what we see on this first green neuron, which, like I said, is a sensory neuron. And because the cell body is located in the peripheral nervous system, it is considered a unipolar neuron. So notice the dendrites will be over here on the tip of the finger. This would be your axon coming down. Then it forms a cell body right over here, and then it continues as another axon and ends on a terminal button. So this green neuron that has the cell body on the peripheral nervous system is your only unipolar neuron. It is true that it will then synapse with another neuron, which we said was the interneuron. But notice how the morphology of this other neuron is actually different, where you don't see a long axon with the dendrites. You already start with this area over here that contains the cell body and the dendrites, then it will have an axon and it will end at the axon terminal. Same thing with the third neuron, which will have the cell body and the dendrites over here, a long axon, and then the axon terminal. So these two, because they are located already in the central nervous system, they are a multipolar neuron. But since they are the ones that are taking the information from the tip of the finger all the way up to the brain, they are considered sensory neurons. So we're going to have two different types of sensory neurons. We're going to have the unipolar neuron, which is the one that starts outside and has the cell body in the ganglia. And then we're going to have either one or two that will be the multipolar neuron that will definitely synapse and start in the central nervous system. So the cell body is what's going to determine if the neuron is located in the peripheral nervous system or in the central nervous system. Once this information arrives in the brain, it will be integrated, processed, and then it will cause an action. And this action is going to come down the motor neurons. And notice how the cell body of this motor neuron is located in the brain, and the cell body already has the dendrites right over there. So this will also be a multipolar neuron. It will then send the information down that will synapse, in this example, with another neuron where the cell body is located in the spinal cord and the dendrites are right around the cell body. Therefore, it's also a multipolar neuron that will then send the action all the way down to the hand for you to sense the temperature of the water. Now, it's important to summarize that the only neuron that is a multipolar neuron is going to be your first sensory neuron, because the cell body of this neuron, again, is located in the ganglia. All the other cell bodies are located in the central nervous system, which can either be the spinal cord or the brain. And because these cell bodies are located in the central nervous system, they're all going to be classified as multipolar. But notice that if it's going up to the central nervous system, all the way up to the brain, it's going to have a sensory function. If it's coming down towards the effector, it's going to have a motor function. We will see in more details these different types of neurons as we move on, but just keep this in mind with regards to their function. So going towards the brain, afferent, sensory. Moving away from the brain and the spinal cord, efferent, motor. And then the positioning again of where the cell bodies are located. If they're located in the peripheral nervous system, unipolar. If they're located in the central nervous system, which can be the spinal cord or the brain, multipolar. Some neurons are going to be named for the histologists who first described them, or for an aspect of their shape or appearance. Examples include the Purkinje cells in the cerebellum, the pyramidal cells that are found in the cerebral cortex of the brain, which do have a pyramid-shaped cell body, and often this very distinctive pattern of dendritic branching will allow identification of a particular type of neuron in the nervous system. With regards to the neuroglias, we have said this earlier, we have two different distributions of these cells that belong to the central nervous system and cells that belong to the peripheral nervous system. The ones that belong to the central nervous system are the astrocytes, the oligodendrocytes, the ependymal cells, and the microglias, and the ones that belong to the peripheral nervous system are the Schwann cells and the satellites. So let's take a closer look at all these cells in the next few slides. This image here is just to show you all the different neuroglias of the central nervous system. We have the astrocytes, which the name basically comes from a star, and because it has all these different projections, it looks like a little star. Then we have the microglial cells, which, like the name says, it's going to be a small for micro, so a microglia. Then we have the ependymal cells, which are these cuboid or these columnar-like cells. And the last one are the oligodendrocytes, which we mentioned already, which are the cells that will form the myelin, which is this lipid substance that will wrap the axons. So let's look at them in more specific details and different functions that they might have. First, we're going to cover the astrocytes, which are actually the largest and the most numerous of the neuroglia. Like I said earlier, they are these star-shaped cells that have these many arm-like processes. As we can see, the processes of these astrocytes, they're going to make contact with other neurons. They're also going to make contact with the pia mater, which is a very thin membrane that will be surrounding the brain and the spinal cord. In addition, they're going to make contact with blood capillaries. So let's look at some of these functions in the next slide. So here we have the functions of the astrocytes. As they are the most numerous of the cells, then they will have more functions. The first one is that they will contain these microfilaments that provide them with this considerable strength, which will enable them to support the neurons. So they form these web-like structures that support the neurons. And since the neurons of the central nervous system must be isolated from various potential harmful substances in the blood, the endothelial cells of the central nervous system blood capillaries, they have very selective permeability characteristics. And the processes of these astrocytes, they're going to wrap around the blood capillaries that secrete chemicals that will maintain this very unique permeability characteristic of the endothelial cells. And in fact, the endothelial cells, they actually create what we call the blood-brain barrier, which will restrict the movement of the substances between the blood and the interstitial fluid of the central nervous system. And details of this blood-brain barrier are going to be discussed later on. In addition, astrocytes in the embryo, they will secrete these chemicals that appear to regulate actually the growth, the migration, and the interconnection among the neurons in the brain. So they serve, for example, with regards to migration, as little pathways for where neurons need to go from one location to the other. Next, the astrocytes, they're going to help to maintain this appropriate chemical environment that's going to be important for the generation of the nerve impulse. So, for example, they will regulate the concentration of important ions such as potassium, the take-up axis of neurotransmitters, and they're also going to serve as this conduit for the passage of nutrients and other substances from the blood capillaries and the neurons. Lastly, we have the astrocytes. They may also play this very important role in learning and memory by influencing the formation of the neuronal synapses. So as we can see, most of these functions, if not all of them, have to do with making sure that the neurons are able to function properly. Next, we have the oligodendrocytes, where oligo means few and dendro means tree. And they will resemble the astrocytes, but they're going to be smaller and they're going to contain fewer processes. So that's where the name comes from. Now, the processes of these oligodendrocytes, like we saw earlier, they're going to be responsible for forming and maintaining this protective covering around the central nervous system axons. And as you will see shortly, the myelin sheath is going to be this lipid and protein covering around the axons that will insulate the axon, and it will also increase the speed of the nerve impulse conduction. And all of this we will see later on, still in this module. Next, we have the microglial cells or microglia cells, and micro means small. Therefore, these are going to be the small cells with these very slender processes that will give off numerous spine-like projections. And unlike other neuroglial cells, which will develop from the neural tube, the microglial cells, they will actually originate in the red bone marrow, and they will migrate into the central nervous system as it develops. Therefore, the microglial cells, they will function as phagocytes. And like tissue macrophages, they will remove the cellular debris that's formed during normal development of the nervous system, and they will also phagocytize microbes and damage nervous tissue. Here we have the last cell of the central nervous system, which are the ependymal cells. Epen means above and dyme means garment. Therefore, the ependymal cells, they're going to be these cuboidal to columnar cells that are going to be arranged in this single layer that possesses the microvilli and the cilia. These cells, they will line the ventricles of the brain and the central canal of the spinal cord, which are the spaces that will be filled with cerebrospinal fluid. Therefore, functionally, the ependymal cells, they will produce and possibly monitor and assist in the circulation of the cerebrospinal fluid. They will also form the blood-cerebrospinal fluid barrier, which will be discussed later. Now we'll be talking about the neuroglias that belong to the peripheral nervous system, which are the Schwann cells and the satellite cells. And both of these, they will completely surround the axons and the cell bodies. As we can see over here, this is the cell body of a neuron. We can see how the satellite cells are going to be surrounding the cell body of this neuron, and the Schwann cells are going to be surrounding the axon of this neuron. Like the oligodendrocytes in the central nervous system, the Schwann cells are going to be important because they are the ones that will produce the lipid in the peripheral nervous system. And the satellite cells, as you can see over here, they surround the cell body of the neuron, and this is a unipolar cell. Remember, the unipolar cell is the sensory neuron that goes, for example, from the tip of your finger all the way to the spinal cord. And where are the cell bodies located? The cell bodies are located on those expansions that are located close to the spinal cord. And those expansions are called the ganglion. So those are where the satellite cells are going to be located. And the main function of the satellite cells are to provide structural support to the neuronal cell bodies, and they will also regulate the exchange of materials between the neuronal cell bodies and the interstitial fluid inside of the ganglia. Like the oligodendrocytes of the central nervous system, the Schwann cells, like I said, they will form the myelin sheath around the axons, and a single oligodendrocyte, as we can see over here, is able to myelinate several axons. But each Schwann cell is actually able to myelinate a single axon. And a single Schwann cell can also enclose as many as 20 or more unmyelinated axons, as we can see over here. And what would be the importance of this myelin? Well, the myelin is important because it will protect the signal that's traveling down the axon as an action potential or a nerve impulse. And can you imagine if we didn't have this protection? As you can see over here, the astrocytes, they're going to be contacting the axon. So this information can get lost in the way. So this is why it's important for the myelin to wrap up the axon and protect that nerve impulse as it tries to reach the terminal button at the end of the axon.